Hello, welcome to BBC Bite Size with the naked scientists, Chris Smith and Kat Arney. You've probably heard DNA being referred to as the genetic blueprint of life, as it carries all the instructions that our cells need. But how is it packaged up in cells and how does it get passed on? So Kat, tell us more.
DNA, or to give it its full name, deoxyribose nucleic acid, is a large and complex molecule carrying the genetic code that tells cells when to multiply, when to die, and what kind of cell to be.
And is everyone's DNA the same?
Not at all. Apart from identical twins, every single person's DNA is unique to them. This is why people can be identified using a technique called DNA fingerprinting. You've probably heard about this being used to settle paternity cases, to identify the father of a baby, or to identify a criminal from samples of cells left at a crime scene. So to make a DNA fingerprint, the DNA can be cut up and separated, forming a sort of barcode that's different from one person to the next.
What about the genes, though? Are they part of the DNA?
Of course. A gene is simply a short stretch of DNA that carries the instructions that tells a cell to make one particular protein. Think of all your DNA as a big cookery book. Each single recipe would represent one gene.
And how much DNA is there in each one of my cells?
Each one of our cells contains more than 2 meters of DNA, packed up into a space just a few thousandths of a millimetre in diameter. This is an incredible feat of biological engineering, and it's only possible because our DNA is wound up into chromosomes.
And what exactly is a chromosome?
A chromosome is a long length of DNA that's coiled up again and again on itself with the help of special proteins until it's tightly coiled enough to pack into the nucleus. That's the special part of the cell where all the DNA is kept.
So just to recap, genes are specific stretches of DNA. DNA is then coiled up to form chromosomes, and the chromosomes are packed inside the nucleus, and that's in the middle of a cell.
That's exactly right. You've probably seen pictures of human chromosomes looking like X's, and we each have 23 pairs of chromosomes, 22 pairs that match, and then a special pair that determine if you're a boy or a girl.
So how does that work?
Well, if you're a girl, you've got 2 X chromosomes, but you're a boy, so you've got an X and a Y. That's how it works for most mammals, but some creatures like birds do have a different system.
Thank you, Kat. So now we know how DNA is packed up inside our cells, and in part 2, we'll discover how it's copied and then split up to make new cells and make babies. Hello, welcome to BBC Bite Size with the naked scientists, Chris Smith and Kat Arney. In part 1, we found out how DNA is packaged into chromosomes within the nucleus of our cells. So, Kat, what happens to the chromosomes when a cell needs to divide?
Normally, cells divide using a process called mitosis. This is essential for life as it allows our bodies to grow, heal and repair themselves, as well as replacing worn-out cells as they die. The end result of mitosis is the formation of 2 new identical daughter cells from one original cell.
And how does it work?
During mitosis, a cell first has to make an exact copy of all its DNA in the form of chromosomes. It's important that this is done correctly, as any mistakes will get passed on to the new cells and could cause problems, which could be very serious. Then the copied chromosomes line up along the middle of the cell.
And then what happens?
Well, the next bit's really dramatic. Microscopic protein scaffolding within the cell pulls on each side of the chromosomes until they pull apart, and a copy of each chromosome goes to each side of the cell. Then the cell splits down the middle and you have 2 new cells, each containing an exact copy of the DNA from the original cell.
Now, we all know that babies are made when a female egg cell is fertilised by a male sperm cell, but how do we actually make those eggs and sperm in the first place?
That's a good question, because eggs and sperm each contain half the genetic information that's needed to make a whole body cell. Essentially, they have 23 single chromosomes instead of 23 pairs. Once the egg and sperm come together during fertilisation, the new cell they create then multiplies over and over again by mitosis to create the millions of cells needed to make a baby. Scientists call eggs and sperm haploid cells, while most of the cells in our body are diploid.
And how does this halving actually occur?
Well, it's all down to a process called meiosis, which is similar to mitosis, but has a few crucial differences. Meiosis only happens in your reproductive organs. In the male, sperm cells are made in the testes, while in female, eggs are made in the ovaries.
So what actually happens during this process of meiosis?
Well, it starts off the same as mitosis. The cell has to make an exact copy of all its DNA, all 23 pairs of chromosomes. But instead of all the chromosomes lining up singly along the middle of the cell, as in mitosis, during meiosis, each matching pair lines up together.
And what do they do then?
Next, one of each pair gets separated into a new cell. So you have 2 cells, each containing one of each chromosome pair. But don't forget that each individual chromosome was also copied right back at the start. So the next step is for these individual chromosomes to line up along the middle of the cell and split apart, as in mitosis.
And what do you get at the end of that?
You're going to end up with 4 cells, each containing half the amount of DNA found in normal body cells, and they're going to contain a random selection from each pair of chromosomes. This is different from mitosis, where you get 2 identical cells, each with a full set of DNA.